Hi everyone, I'll be talking today on autoimmunity. Autoimmunity means immunologically competent cells in one's body react with the self antigens and produces the antibodies, and these antibodies results in producing certain diseases. All these will be considered under the topic autoimmunity, and today's in this class I'll be talking on what is immunological tolerance and how the breaches will occur in an immunological tolerance, and how this breaches in immunological tolerance results in autoimmunity. So let's move on this particular topic. So if you see, the central function of the immune system is to distinguish foreign antigen from the self antigen. So always the foreign has to be differentiated from the self antigen, so they won't bound any immunological response. So once the self antigens are reacted with the immunologically competent cells, they bound the immune response, results in disease. So the self antigens usually are not immunogenic, but once they become immunogenic, they will cause for the production of their concerned antibodies, which results in the autoimmune disorders. So this particular property, that is the self antigens won't react with the self T lymphocytes, is known as tolerance. So loss of this kind of tolerance, this kind of immunological tolerance, leads to mount an effective and specific immune response against the self antigens, and this results in autoimmune diseases. So this is what the mainly it involves autoimmune production of autoimmune diseases. So this autoimmune diseases produced when the self antigens get reacted with the self T lymphocytes or T cells. So this is what the important point we have to remember. The next one is, if you see the historical introduction about the aspects of this particular autoimmunity, Paul Ehrlich in 1901 first time demonstrated the antibodies against the RBCs in the goat when the one goat received the RBCs from another goat, and he named it as horror autotoxicus. So even though the goat related to the both goats are related to the same species, the RBCs from one goat when introduced into the another goat are responsible for severe hemolysis, and it was named as horror autotoxicus. That was the name first time given for this particular events. And Metalnikov demonstrated that sperm immobilizing antibodies in the guinea pig. When the spermatozoa obtained from the guinea pig and introduced into the same guinea pig, he demonstrated the antibodies against this guinea pig spermatozoa. And Donath and Landsteiner identified that autoantibodies against the RBCs among the people who are suffering with paroxysmal cold hemoglobinuria, and he named those autoantibodies against the RBCs are named as hemolycins. So these are some of the historical aspects of this particular autoimmunity. Then immunological tolerance, how the individual, I already mentioned that what is the meaning for immunological tolerance. Immunological tolerance means non-responsiveness of our immunologically competent cells against our own cells is known as immunological tolerance. So how this immunological tolerance will be attained? There are two important things are there. One is central tolerance, another one is peripheral tolerance. First we will see the central tolerance. Central immunological tolerance will be obtained by the deletion of self-reactive T and B lymphocytes while these cells are getting matured in the central lymphoid organs like in thymus and also in the bone marrow. When this T and B lymphocytes before getting matured into the immunologically competent cells, they produce from the bone marrow, enter into their concerned lymphoid organs like thymus in case of T cells and bone marrow in case of B lymphocytes. So when these cells, immature T lymphocytes enter into the thymus, By means of apoptosis, all the unwanted self-reactive antigens will be removed, and that means the T lymphocytes will get educated not to react against the self-antigens. And B cells in the bone marrow, by means of antigen receptor editing, they will get educated not to respond against the self-antigens. Then peripheral tolerance, this is a main thing when compared to the central. The first one to be remembered in the peripheral tolerance, immunological tolerance to attain, first one is ignorance, the property of ignorance. Ignorance means the self-reactive T cells will ignore the self-antigens so that the immune response will not be bounded, so antibodies will not be formed, and there is no possibility for any kind of autoantibody related diseases. Another important thing is energy. Energy means unresponsive or non-responsive to antigenic stimulus. First one is, before going to understand about this one, first we will see once the T cells gets activated, what will happen. See, whenever the antigen presenting cell, this is the antigen presenting cell, this antigen presenting cell will present the antigen to the T lymphocytes, through the MHC complex. Okay, through the MHC complex, it will be presented to the T cells through the T cell receptor. So through the T cell receptors, the antigen will be presented by the antigen presenting cell to the lymphocytes for the activation. So once the antigen is presented to the T lymphocytes to the TCR, apart from the T cell receptor activation, the co-stimulatory factors B7, the B7 on antigen presenting cell will get reacted with CD28 on T lymphocyte. This is also known as co-stimulatory effects. So not only the attachment of antigen to the T cell receptors is not enough to activate the T lymphocyte, and also the B7 receptor on the APC has to be reacted with the CD28 on T lymphocyte. Then only it may result in the production of useful cytokines to bound the immune response. But what will happen in case of energy, that means unresponsive T antigens, when the self antigen is presented to the T lymphocytes, when the self antigen present to the T lymphocyte through the antigen presenting cell, the co-stimulative factor, that is B7 on the antigen presenting cell, will react with the CTLA4 on the T lymphocytes in spite of reacting with CD28 on the T cell receptor, T cell surface. So this kind of co-stimulatory effect will result in non-responsiveness to the antigenic stimulus, and so it won't produce any kind of immunological response. Then phenotypic skewing, that means the phenotypic skewing, that is T cells react with self antigen, and when the T lymphocytes react with the self antigen, results in non-pathogenic cytokines. You all know that when the T cells are getting sensitized, or when they got activated, the cytokines will be produced. But here, once the T lymphocytes are activated with our own antigens or self antigens, results in the production of non-pathogenic cytokines and chemokine receptors, so that immune response will not be bounded. And next one is, I think this is 3, and next one is fourth one, that is apoptosis by apoptosis by activation of the induced cell death. See what will happen here again, you can see here, once this antigen is presented through the antigen presenting cell to the T lymphocyte through the TCR, that is a T cell receptor, the co-stimulator factors B cell will react with the CD28 on T lymphocytes, and this reaction will result in the expression of FAS ligand, FAS ligand on the T cell surface, which reacts with the FAS receptor FAS receptor on the T lymphocytes. So it results in death of the T lymphocytes which activated against our own cells. That is apoptosis of the self-reacting T lymphocytes by the activation of with the self antigens. And next one is T regulatory cells. These T regulatory cells will down regulate the self-reactive T lymphocytes when they get exposed to the self antigens and produces interleukin 10 and transforming growth factor beta. These two will down regulate the immunological responses produced by the T lymphocytes when they got exposed to the self antigens, and so the immunological response will not be bounded. And next one is dendritic cells. These dendritic cells again down regulate the co-stimulatory ligands like CD40 and B7 on the on their cells and causes for non-bounding of immunological response. And seventh one is sequestration of self antigen. Some of the antigens will present in some areas where they never exposed to the immune system of the host, like lens protein, which is present in the lens capsule of the eye, and spermatozoa by the time won't be produced, and brain cells, and these won't cell, all these cells will be hidden in various parts of the body, and they never exposed to the self-reactive T lymphocytes, so that there is no possibility for bounding immunological response. By all these means, the individual will attain the immunological tolerance against the self antigens. So the contributing factors for the loss of this immunological tolerance, one is genetics, and environmental factors are there, immunosuppressive therapy, and varieties of infections produced both by bacteria as well as bacteria, viruses, and even parasites. All these infections may cause for the loss of immunological tolerance, results in the bounding of autoimmunity. Then Ernst Witebsky criteria, this means just like our Cox postulates in bacteriology, what we talk about, same way Ernst Witebsky proposed certain criteria to say that one particular disease is produced by one particular disease is of autoimmune etiology. So in any case of disease, if you would like to stamp it or confirm it as an autoimmune disorder, in that particular individual, both antibody mediated immune response and cell mediated immune response are individually either antibody mediated or cell mediated immune response have to be activated. Then responsible antigens should be identified, isolated, and should be characterized. And third one is immunopathological changes by the same antigen should be induced even in the experimental animals also. And fourth one is, since it is mediated through the antibodies, passive transfer should be possible. So then only we can say that that would be of autoimmune etiology. Then what are the features of autoimmune disease? One thing is there should be autoantibodies against the self antigen, and in the lesion produced in case of autoimmunity, the lesion should be infiltrated with the lymphocytes and plasma cells and also the immunoglobulins. And third one is the response. Always the autoimmune disorders should be respond against the corticosteroid treatment and immunosuppressive agents. And there should be a history of genetic predisposition, and there should be more than one disease. Usually autoimmune disease should not be present isolatedly. Most of the times they present with more than one disease in a particular involved individual. And what are the mechanisms of autoimmunity? We learned that immunological tolerance is responsible for not bounding the autoimmune disease. But once this immunological tolerance is breached up or breakdown, then it results in autoimmune diseases, which is known as autoimmunity. So what are those mechanisms involved in breaching of immunological tolerance? First one is release of sequestered antigens. We already mentioned that there are certain sequenced antigens which are present in some primary areas like lens protein and spermatozoa. If you see the lens protein, usually it's present within the capsule in the eye, and followed by the cataract surgery or followed by injuries to the eye. These lens proteins will enter into the circulation. Even before that, the lens protein was not exposed to the immune system to recognize it as self-antigen. It's the first time it's getting exposed to the immune system. So the immune system will react against the lens protein and produce the antibodies. And these antibodies are produced against our own lens protein. So they are known as autoantibodies. And these autoantibodies will damage the eye by producing the autoantibodies. Next one is spermatozoa. Usually the spermatozoa present in the seminiferous tubules or seminal vesicles, but followed by the disease like mumps. It's a viral disease common in childhood, and this mumps disease will cause for the damage to the basement membrane of the seminiferous tubules. And once it happens, the spermatozoa released into the circulation. Once it's released into the circulation, the antibodies are produced against the spermatozoa. Antibodies are produced against the spermatozoa, and these antibodies again acts on the spermatozoa and forms immune complexes, results in the antibody mediated arthritis. And this arthritis sometimes may result in sterility also. It may cause for sterility in that particular individual, right? This is about the autoimmune mediated arthritis. And next one is the new antigen formation. New antigen formation or antigenic variation on the cell. When certain cells like RBCs and platelets, especially when they get exposed to certain chemical agents or certain drugs, are followed by irradiation, or when the people suffered with the viral or bacterial infections and the mutation of the cells, the antigenic nature of surface antigenic nature of these cells get varied, will altered. Once the antigenic structure altered, these cells will be recognized as new cells in the body, and then against which the T lymphocytes gets activated and causes for antibody formation. This antibody results in the various damages. Next one is polyclonal B cell activation. Means multiple B cell clones will be produced followed by certain infections. So these multiple or polyclonal cells means the cells which are clones which are produced non-specifically against antigenic stimulus. So antigenic non-specific stimulus of the B lymphocytes produces many number of unwanted multiple B cell clones which can produce their concerned antibodies. That is known as polyclonal B cell activation. This is possible in case of tuberculosis and followed by the treatment with nystatin, and viral infections like Epstein-Barr virus and mycoplasma infections and parasitic infections like malaria. All will non-specifically will stimulate the B lymphocytes to produce many number of clones which can produce many number of unwanted B lymphocytes, antibodies, which results in breaches in the immunological tolerance to bound the autoimmune response. And next one is molecular mimicry. Means the identical peptide sequence present among the epitopes. Epitope means antigenic determinant on a cell or tissue. So identical peptide sequence among the epitopes of microbes and self-antigens. So on the microbes and on cells, these particular what we call the antigenic determinants on this in a cell and on the bacteria, and both will be on the bacteria, both will have the identity, and this identity will cause for bounding of immunological response. Say for example, Shigella species are having the identical sequence of the BLA27, and Mycobacterium tuberculosis and synovial membranes of the joints will have this kind of molecular mimicry, and Coxsackie B virus and the myocardium in the individual. So in, say for example, if you take one example, the Coxsackie B virus antigens produces the antibodies. It produces the antibodies, and these antibodies, the antibodies which are produced against the Coxsackie B virus, these antibodies will act on the myocardium of the infected individual. Why? The reason is the Coxsackie B viral antigenic structure similar to the myocardial antigenic structure. So the antibodies which are produced against this particular virus will recognize the myocardium as their antigen, and it reacts with the myocardium of the host, results in myocardial damage. The same thing in case of Mycobacterium tuberculosis, and same thing in case of Shigella flexneri. Next one is cross-reacting antigens. The same type of antigen, organ-specific antigens are present among the different species. For example, sheep brain vaccine. We have many number of vaccines against the rabies. Anti-rabies vaccine, which is prepared from the sheep brain tissue, has got the structural similarity with the brain here. Yeah, I mentioned this. Structural similarity with the brain cells of the receiver, that means human beings. So the sheep brain cell antigens are almost similar to the human brain antigen. So when you receive the anti-rabies vaccine, which is prepared from the sheep brain tissue, that vaccine not only consists of the vaccine particles but also contains the sheep brain particles, because it's being prepared from the sheep brain tissue. So once we give this injection to the individual, to the recipient, then not only the antibodies against the rabies virus produced, but also the antibodies against the sheep brain tissue also will be produced. And these antibodies, the sheep brain tissue antibodies will recognize the human brain cells as their antigen because of antigenic similarity, and bounds the immunological response, causes for damage. Like this here, you can see here the brain cells. These are all the antibodies. These are the antibodies. These antibodies will recognize the brain tissue, the neuronal tissue as their antigen, cause for the damage. And next one is exposure of forbidden clones. Means during the developmental stages of the T and B lymphocytes, some of the epitopes on the cells, or the cells will not be removed totally, and these forbidden or hidden clones can get exposed during the lifetime and causes for the production of uncontrolled number of antibodies. And therapy for certain conditions, like when you give the treatment in certain conditions, like especially cancerous conditions and other conditions, they can modify the T cell function and causes for the autoantibody formation. Say for example, if you use the cyclosporine, which can alter the T cell proliferation. If you use the corticosteroids, they can inhibit the function of T lymphocytes, and if you use the cyclophosphamide, they can kill the T lymphocytes, and you can use the monoclonal antibodies, and these monoclonal antibodies acts as an antagonist for pro-inflammatory cytokines or co-stimulatory molecules and the T cell and antigen receptors, and causes for the modification of the T lymphocyte. Once the T lymphocyte activity is modified, then it may result in bounding of immunological response against our own antigens or self antigens. Next one is macrophage dysfunction or otherwise called the antigen presenting cell dysfunction, also results in this breaches in the immunological tolerance, and defects in IR genes, that is immunoregulatory genes, IG genes, that is immunoglobulin genes, also will cause for this particular kind of breakdown of immunological tolerance, which is responsible for binding of or bounding of autoimmune response. Then autoimmune diseases, there are varieties of diseases are there. One is, first we will discuss about organ and cell type specific autoimmune disorders. Say for example, in case of autoimmune hemolytic anemia or drug induced autoimmune hemolytic anemia, the erythrocytes will get affected. And here in this case, the self antigens are produced against RBC membrane protein, and drug induced like the penicillin and methyl dopa, once they act on the erythrocytes, they cause for alteration of the antigenic surface antigens on the RBCs, and then altered antigenic surface RBCs will be recognized by the immune system as foreign cells and cause for the production of autoantibodies, results in autoimmune hemolytic anemia. Next one is pernicious anemia. In case of pernicious anemia, the intrinsic factor, the antibodies are produced against the intrinsic factor and results in anemia. So like this, autoimmune disease related to the RBCs are autoimmune hemolytic anemia due to the drugs, or pernicious anemia due to the antibody production against intrinsic factors. Next, in case of platelets, idiopathic thrombocytopenic purpura is the disease where the autoantibodies are produced against platelet membrane protein. And next one is in case of good passage syndrome. This disease affects the kidneys and lungs, and where the self-reactive T lymphocytes acts against the renal glomeruli and alveoli of lung basement membrane, and results in the formation of autoantibodies, results in damage of those particular areas. Next, in case of Graves' disease, thyroid gland is involved. Here, the thyroid stimulating hormone receptors acts as a self antigen against which the antibodies against the thyroid stimulating hormone receptors will be produced, and which are known as LATs, long acting thyroid stimulator. And this autoantibodies are responsible for damage to the thyroid gland tissue, results in the disease. Another important disease related to the thyroid gland is Hashimoto's thyroiditis. Here the self antigens are thyroid proteins and various cells. So here also the autoantibodies are produced, results in damage to the thyroid gland. Like this, various varieties of self antigens present in various types of cells in the body, like RBCs, platelets, kidney cells, lung cells, and thyroid gland cells, results in these self antigens, when they get exposed to the immune system of the host, the immunological response will be bound and results in either anemia or good passage syndrome or Graves' disease or Hashimoto's thyroiditis. Next one is organ specific autoimmune disorders. Next one is the pancreas related autoimmune disorders. This is known as insulin dependent diabetes mellitus, where the autoantibodies are produced against pancreatic B cells, and here the T helper cells also plays a major role, and autoantibody also plays a major role to produce the disease in the pancreas. Next one is myasthenia gravis. Here the self antigens responsible for myasthenia gravis are acetylcholine receptors, against which the autoantibodies are formed. When the acetylcholine receptors are got exposed to the self-reactive T lymphocytes, causes for the autoantibody formation and results in myasthenia gravis, which is related to the skeletal muscle of the body. And next one is post-streptococcal glomerulonephritis, which affects the kidney. Here the self antigens are basement membrane antigens. So the antibodies which are produced against the streptococcal antigens, and these antibodies acts on the basement membrane of the glomeruli of the kidney, and forms the antigen antibody complexes, and this complex mediate antigen antibody complex mediated immunological damage will occur. And in case of spermatozoa, where it acts as an self antigen, and here again this was discussed already, the autoantibodies are produced when the spermatozoa enters the circulation, results in male sterility by damaging the testes, which is known as autoimmune mediated arthritis. And next one is systemic lupus erythematosus, where the autoantibodies are produced against the DNA of the cells and the nuclear proteins, RBCs and platelets in the body, and causes for the systemic lupus erythematosus. And then next one is related to the lower spine, that is ankylosing spondylitis. Here the self antigens are the sacroiliac joints, the membrane cells, synovial membranes present in the sacroiliac joint and other vertebra, and here the autoantibodies which are produced against these antigens known as autoantibodies. This autoantibody immune complexes causes for damage to the lower spine, result which is known as ankylosing spondylitis. Next one is in case of multiple sclerosis, the self antigens here are brain or white matter of the brain, and the self-reactive T lymphocytes produces an inflammatory lesion which destroys the myelin, in brain or white matter of the brain, results in multiple sclerosis. So here the affected tissue is myelin of the central nervous system. Next one is in case of scleroderma. Here the self antigens are nuclear antigen of the heart, lung and GIT and also the kidney. So when these antigens exposed to the immune system, then the self-reactive T lymphocytes produce antibodies against all these cells, results in the connective tissue disorder in the form of scleroderma. So here one important point you have to remember is, unless the T cell gets activated, only B cells are not able to bound the autoimmune response. Always the T cell activity is required to produce the autoimmunity. T cell independent autoimmune disorders are rare and almost all they won't produce. So the T cell, self-reactive T cells are very important to bound the autoimmunity, even though it is antibody mediated one. And next one is Sjogren's syndrome. This Sjogren's syndrome, the autoantibodies are produced against ribonucleoprotein antigen SSA and ribonucleoprotein SSB present mainly on the salivary glands and also lacrimal glands. Salivary and lacrimal glands presenting this ribonucleoprotein antigen SSA and SSB, against which the antibodies are produced, responsible for Sicca syndrome or dryness of the mouth and dryness of the eyes. It's also known Sjogren's syndrome, otherwise known as Sicca syndrome. So mainly here it affects lacrimal ducts. Of course, these ribonucleoproteins also present in the liver, kidney and the thyroid also. So related disease also may be present in that particular individual. Then SLE, if you see the SLE, which is already I mentioned about the SLE in the disease process or mechanism of this disease. In the SLE, which is a fatal, chronic and multi-systems involved disease, where you can see the multi-systems are involved here, and mainly it causes for the nephritis, which is known as lupus nephritis, and pleural effusions also you can see, and heart problems you can see, and joints involved, and results in arthritis, because the autoantibodies are produced in the nucleus of the various cells against the DNA of various cells, results in all these clinical manifestations. And next one is a Raynaud's phenomenon, also you can see here in SLE. Raynaud's phenomenon we can discuss in later, or to know about when the fingers are get exposed to the extremes of cold, then it results in cessation of the blood supply to the fingers and results in gangrenous like this. So this Raynaud's body phenomenon also seen in case of SLE. And other symptoms of SLE may vary differently, but you can see the butterfly rash. you can see the butterfly rash. See, this is the butterfly rash on the face of infected individual, you can see. So this is a very common thing which we can see in the females. That's why I focused here the female photograph. And next one is for the diagnosis, anti-DS double stranded DNA antibodies is more specific, and anti-nuclear antibodies also can be done, but it's non-specific test. But preliminarily we will do the ANA detection. If the ANA is positive, then only we can go for anti-DNS, double stranded DNA antibodies to diagnose this particular condition. And the next important thing is rheumatoid arthritis, and which is a systemic polyarthritis. Many number of joints are involved with muscle wasting and with the formation of subcutaneous nodules. These are all mainly due to the serositis and myocarditis and vasculitis. All these inflammation of the vasculature, inflammation of the heart muscle, inflammation of the synovial membrane of joints, all these things you can see in rheumatoid arthritis. And here what will happen is the autoantibodies are produced against host immunoglobulins antibodies, host immunoglobulins, and these antibodies against the antibodies is known as RA factor. This IgM type directed against the FC region of IgG. So here in case of rheumatoid arthritis, IgM type of antibodies are produced against IgG antibody. Okay, is it clear? Don't get confused. What is this antibody and antibodies against antibody? So the antibodies, we have the antibodies in our body. So against our own antibodies, IgM type of antibodies are produced, and that is the specialty of this rheumatoid arthritis. And here we can see the infiltration of the synovial membrane with the lymphocytes. See, this is the synovial membrane. In this synovial membrane, you can see the infiltration of the cells. See, these are all the inflammatory cells. These inflammatory cells are getting infiltrated like lymphocytes, plasma cells, and antigen presenting cells. All these are present on the articular surface of this synovial membrane, which is known as rheumatoid pannus. And because of formation of this particular rheumatoid pannus, usually people will suffer with severe pain. So the important thing here to remember is we have to always, rheumatoid arthritis has to be differentiated from osteoarthritis. In case of osteoarthritis, the pain will be present in case of activity. With the activity, the people will feel pain. But in case of rheumatoid arthritis, even pain is present in rest. During the rest also, the people will feel pain and swelling of the joints, and painful inflammatory or swollen joints are seen in rheumatoid arthritis. But in case of osteoarthritis, if the people take rest, the pain will be subsided. That's the most important point to remember to discriminate both the rheumatoid arthritis and osteoarthritis, because both of them most of the time present similarly clinically. Then lab diagnosis, varieties of tests are available. One is the latex agglutination test, but it's not much reliable, and anti-nuclear antibodies we can do, but more specific test to diagnose this condition is anti-citrullinated peptide antibodies. Anti-citrullinated peptide antibody test is more specific to diagnose this particular condition. And the next one is the involvement of these varieties of joints. See, in case of acute condition, in case of acute rheumatoid arthritis, you can see the joints, swollen joints with the red inflammatory joints. See, these two joints inflamed and red in color. See, these are all, here also you can see small joints are involved. All these joints are inflamed. Here also you can see the inflamed joint spaces. But in later stages, because this rheumatoid arthritis will be seen in many number of episodes, in each episode will be shown like this, and after some period, because of many number of episodes, the joints will be flexed like this. See the hand, how the hands of rheumatoid arthritis I mean chronic rheumatoid arthritis patients are seen. Here you can see many number of, you can see the very commonly involved joints are frequently affected joints, or see the elbows. Less commonly affected areas are elbows, and more commonly affected areas are small joints of the hands. See the small joints of the hands and also the legs. Small joints of legs are involved, even including the ankle and the wrist also gets involved. But elbow joints and the hips and neck, here the elbow will not get infected. Hip joints and also the neck joints won't get involved, and the hip joints also won't get involved in case of rheumatoid arthritis. And another important thing is autoimmune disease which are associated with the MHC or major histocompatibility genes. Rheumatoid arthritis associated with the DR4 HLA gene, and insulin dependent diabetes mellitus associated with DR3 and DR4. Multiple sclerosis and good passes associated with DR2 alleles of HLA gene, and SLA and psoriasis and IBS, that is infective bowel syndrome, irritable bowel syndrome. These are again associated with the DR2 and DR3, and ankylosing spondylitis associated with the B27 HLA gene, and Graves disease associated with the B8, and celiac disease associated with DQ2 and DQ8. These are all the association between autoimmune diseases and MHC genes. That means the people who shows this HLA gene will prone for this kind of autoimmune disorders. Then treatment, once the disease is initiated, it's self-perpetuating. It continuously goes on. So we have to give the immunosuppressive agents and corticosteroid therapy also to be given, and plasmapheresis can be done. Plasmapheresis means the plasma from the body, the blood from the body removed and passed to through a machine, and where the all unwanted auto antibodies will be removed. Auto antibodies will be removed, and so that we can reduce the severity of condition. And vaccination with the T cells, they are not much properly being used, anyhow available, and administration of synthetic peptides. Among all, we mainly depending on immunosuppressive agents and corticosteroids to treat this autoimmune disease. And take home message is autoimmunity is an aberrant form of immune response due to loss of tolerance to the self antigens. And next important point is autoantibodies are formed as a result of infection and tissue injury, and autoantibodies are responsible for lysis of the cells and formation of immune complexes and sensitization of T cells, which results in disease either by means of damage to the cells or due to damage to the cells by means of antigen antibody complex formation. That is nothing but type 3 hypersensitivity type of reaction. And all these causes for the disease manifestations, and treatment, corticosteroids and immunosuppressive agents have to be given. And regarding this autoimmunity, it's a time to remember, this is time to reality and time to heal. That means to diagnose this condition, it will take some time, and once you diagnose, it will take some time also to get healing from that particular disease. So autoimmune disease, after excluding all other possibilities, will go for the autoimmune test. That's why it will take some time to know the reality and to get treatment also. It will take some time. That means to get healing from the disease also, it will take some time. This is about the autoimmunity, and thank you very much.